Hello everyone, welcome to the RNA collaborative seminars. My name is Gian Tartaglia from the Italian Institute of Technology, Italy. I have the honor today to chair this section. We have two speakers, Iuliia Kotova is the first one today. As a quick introduction to Iuliia, she defended a PhD on Influenza A virus in the lab of Gian Kosinski, and the lab is interested in understanding um, how viruses and parasites function using structural and system biology approaches. So, Iuliia, the floor is yours, and have a nice one.
Thank you. I just will share my screen. Yes, today I will tell you how the influenza A virus exploit phase-separated organelles, specifically paraspeckles, and why does it do that? Oh, sorry. Didn't share it.
Yeah, you can see it.
But yeah, okay, now it's working. Thank you. So I first, of course, want to introduce influenza A virus. It is also RNA virus, and it has segmented genome. That basically means that each of its gene has its own polymerase complex, and then RNA is embedded in the multiple copies of NP protein. Then influenza also encodes of a couple of other proteins that help it infect the cell. And interestingly, for the RNA virus, when influenza infect and goes inside the cells, it developed the strategy to go inside the nucleus and replicate there. And this is quite unusual because, as a rule of thumb, the DNA viruses replicate in the nucleus, while RNA viruses prefer to replicate in the cytoplasm, because otherwise it's evolutionary expensive to develop the strategy to cross the nuclear barrier, so nuclear envelope, and then upon production of the genetic material to go to deliver it back to the cytoplasm. Nevertheless, influenza does it, and we were interested in why does it do it and what kind of interaction does it undergo inside the nucleus. And there are several ways to study interactions of the viral host proteins, but we were specifically interested in the most native conditions. So we wanted to study this interaction via in-cell cross-linking. And the only question was why the people didn't do it before. Influenza is not a new virus. It is for more than 100 years with us. And the reason is that I already mentioned influenza only has 8 genes, basically resulting in a bit more than 10 proteins. And even though viral infection is usually, in principle, overexpression, the cell is done of the protein. So the human proteins will always compete the viral ones. And if we're doing cell cross-linking just like that, we will never identify enough viral host pairs. However, influenza itself, as many other viruses, already give us some clue how to make it more efficient. So many viruses have a program so-called host shutoff, where they specifically can downregulate the host expression, so the translation of host proteins, and favor the translation of their own ones. So we hypothesize, so if we would find a way to enrich for newly synthesized protein upon infection and then cross-link, that the majority of the pairs that we should have would be then at least containing a viral protein, because it would be cross-linked. And indeed, this is what our colleagues in Berlin, Van Leo and Boris Bogdanov, did. So this is our workflow. At the beginning, we just infected the cells with the influenza virus. We waited till the infection will start going, and when the host proteins would be downregulated in the translation sense, and then we exchange one of the amino acids with the synthetic one, would give it time to label, and then we just normally cross-link, lyse the cells. And here we can add an additional step, because now we could, through this synthetic amino acid, we now could enrich specifically just for the newly sensitized proteins, in our case, meaning mostly for the viral proteins. And then just a normal standard mass spec workflow. And it did work. We have identified over 100 viral host protein interactions, but it's still very complex graph. So in order to understand where these interactions takes place, we map them in already published a map of the human organelles. And here in orange, you can see the host proteins that are interacting with the influenza proteins. And when we did enrichment analysis, we noticed that the majority of these interactions are taking place in so-called nuclear bodies. And upon refinement, we could see that it's not just some, or many nuclear bodies, not one organelle, but all of these interactions were mostly enriched in a small organelle that is called paraspeckle. And paraspeckles are membraneless organelles based through phase separation, and they always contain long non-coding RNA NEAT1, and then several other essential and not essential proteins, including SFPQ, non and PSPC1. They are highly similar to each other. And they were identified more than 20 years ago. However, by now we still don't know what are their precise functions. They were shown that maybe they're involved in the gene regulation. They seem to be associated, though, with stress. And of course, viral infection is always stress for the cell. So it become very interesting to see why there are so many interactions between viral and paraspeckles proteins. We first zoom in our network. So we see here again in orange, these are two of the viral proteins, so nuclear and non-structural protein 1. And they indeed interact with many paraspeckle proteins, and several of these proteins were essential. So the one that are in pink, so essential means without these proteins, paraspeckles cannot actually be built in the cell. We first, of course, cross-validated this. We just did a native IP and could see that indeed this protein here, nono, could we could pull down its known interaction factors, but also NP and NS1 were among the most enriched proteins. But of course, we were also interested what happens on the organelle level, not just on the protein interaction level. And the easiest way to understand what's going on with paraspeckles is to stain using fish for NEAT1. And we did. We can clearly see paraspeckles here in the non-infected cell. However, when the infection progresses, they seem to disappear. So already after 8 hours post-infections, there were basically very few paraspeckles present, and they were all significantly smaller. And for us, the question was raising, okay, how does it happen? How we come from the intact paraspeckle here to almost non-present already by 12 hours post-infection. And because we've seen that there are two viral proteins that have many cross-links to the paraspeckle proteins, we first overexpressed them. So here's just a GFP control to see that the overexpression alone does not cause disassembly of paraspeckles. However, overexpression of the viral proteins, so here we see NP positive cells do not have any paraspeckles presence. The same is true for NS1 positive cells. And as nuclear protein is also present in the cells, not only in the free state, but also as a part of viral ribonuclein complex, we also have overexpressed those together. And after quantification, we noticed that VRNP has actually stronger effect than NP alone. And we first were puzzled by this, but then we found that we maybe know the answer why VRNP has a stronger effect than NP alone. And when we look at the polymerase complex of influenza, we have one protein, PA, that through a frame shift during translation builds a separate isoform, PIX, and this isoform is only having endonuclease domain. And this domain helps also influenza to downregulate the host genes because it's there to degrade the host mRNA. We don't know the mechanism, how it can differentiate between viral and host mRNA, but it definitely can. It was already shown before. And we also know the binding motif of PAX, and we could see also that NIT1, though not shown before as being a target of PAX, has this binding motif. Moreover, other long non-coding RNAs were also shown before to be targeted by PAX protein. So in the next step, we just overexpressed specifically this PAX isoform, and we could also see that the cells that are positive for these proteins also don't have paraspeckles. Now we already have two different mechanisms that seems to be contributing to the disruption of paraspeckles. On one side, we have viral proteins NP and NS1 interacting with the paraspeckles components and causing dissociation. On the other side, we have the endonuclease that is degrading RNAs. So when we look here, we see that the paraspeckles are disrupting between 4 and 8 hours post-infection. However, when we did the qPCR of the NIT1, because here it might be that the NIT1 is still present 8 hours, but we just cannot see it because it's diffused in the cells and not highly concentrated anymore. And it seems to be indeed what is happening, because up to 12 hours, we still see more or less stable levels of NIT1, while we cannot already see intact paraspeckles anymore. So what we think is happening is actually that the primary cause of paraspeckle disruption is not PIX. So in our model, we see at the beginning of the infection, we have the paraspeckles that are functioning and present. Then when the infection progresses, there is more and more viral proteins present around, and they start interacting with the paraspeckles components, probably causing dissociation and also leaving NIT1 unprotected. And then the PIX can degrade it much easier. That's why after 12 hours post-infection, we don't see also the NIT1 present. But also, I haven't shown the data, but we know from literature and our own data that at the end of the later phases of influenza infection, there is no, the polymerase 2 gets stalled, and there are several papers of before that shown if the POL2 gets stalled, the paraspeckles also start disassembling. So even if the first two, of course, this mechanism happens later, but it might still a bit contribute at the later stages of infection. That's why we don't see paraspeckles upon infection. But we are working with virology. For us, is of course also very interesting why the virus would bother, why the virus would do that. And to answer this question, we downregulated with RNA interference several paraspeckles, paraspeckle proteins to see and then we infected the cells to see what the effect would have on the infection. And here we're first very confused because we have here some proteins upon downregulation of which we see the increase in the infection, while we have then they play a restrictive role in AIV biology. And then we have proteins that various SFPQ seems to be also shown before in the literature that if we downregulate this gene, the infection cannot proceed this efficient. So it has a proviral function. And then we have several proteins that do not seem to play any role. And we thought that there should be some duality in this phenotype, and to see, because the SFPQ was shown before with this time, and it's very difficult to work with the NIT1 because it's very long. We focused on NONO. because after NIT1, it has the strongest phenotype in restrictive the infection. So at the beginning, we first generated knockout cell line and just replicated our phenotype with knockdown. The same, upon knockout, we see the increase in the infection. And then we generated the cell lines with the rescued nono. And we could rescue the phenotype for one clone partially, for the other one fully. And also here we can see reversely to the knockout of NONA. Upon overexpression of NONA, even in the wild type cell, we see also the decrease in infection. We also measured the titers of the virus to see how fit the virus is upon passaging. And we can see that indeed the virus grows best in the cells that do not have NONO present and worse in the cells that overexpress NONO. And why we think it's happening, as I showed before, that there is a double phenotype between there are genes that increase infection, the one that decrease. So as to understand how the interaction happen inside paraspeckles, we did IPMS of NONA under mock and in infected condition. And here we see on the left side the non-interactors in non-infected cells, and in these all colored dots, NP and NS1 interacting partners. So there is a lot of NP and NS1 interacting partners interacting with NONO, though only under non-infected conditions. And that means that upon infection, these interactors get released. And we think this is the explanation of why influenza bothers to disrupt paraspeckles. So we think that through different mechanisms, through direct interactions with NP and NS1, through activity of its endonuclease protein, and through the inhibition of POL2 late in the infection, influenza virus actually disrupts the paraspeckles in order to release some other proteins such as SFPQ that are necessary for to enhance the viral replication. And I think I actually already done. I was a bit faster than I expected. And yeah, this is already a preprint. If you want to read a bit more about this, you can definitely check it out. And of course, I would like to thank Gian and whose lab I was able to do this work, and all my colleagues and collaborators who participated in this project. Thank you.
Thank you very much, Iuliia. It is a very fascinating story, actually quite broad, I would say, in terms of direction that can be taken to further investigate the uh, the role of influenza type A and in the disassembly of paraspeckles. I don't see for the moment questions, but I might have one maybe while we wait a little bit. I see, especially in this last slide, that you showed that there are parallel mechanisms, especially for the case of FFPQ. So in a way that um, the knockdown will be, you know, in favor of stopping the replication of the virus. I wanted to ask you if all these layers, including PAC-X, are somehow synergetic, so in a way, so they run in parallel, or they are, you know, mechanisms that you see as independent, separated. So in other words, um, um, the um, the same proteins could be perhaps involved, so the ones of the paraspeckles involved in more interactions that could be relevant for the virus. I don't know which of them, in your opinion, is more important, or if there are several mechanisms that could be in place for the um, disassembly and replication of of the virus. Also, I find very interesting you say that some proteins are liberated to be participating in the reproduction of the virus, and whether if you have idea which of them could be more important. Well, you made an example, of course, but maybe there are also others that are interactors of interactors or things like this. I don't know if you want to give us a little bit of. I find it very nice, very inspiring story.
Yeah, because it's like we did not focus on because we know that in the paraspeckles there are at least two proteins that are absolutely necessary for viral replication. On one side is SFPQ that I mentioned. There the function are not clear, but we know that replication transcription of the virus specifically is then going very slow if we don't regulate it. But for the other protein, I also didn't mention it because there are already several other studies for HNRNPK that it's necessary for the splicing. There are two proteins inside influenza, NS1, NS2, and M1, M2 that get spliced. And this protein that is also essential for paraspeckles is also essential for splicing of the viral proteins. And we think that it's also the release of this protein necessary to support viral replication. And of course, they're also in the low dosage, they're present in the nucleoplasm, but like a lot of these proteins is concentrated inside paraspeckles and probably just faster for a virus to get it out in the free form.
I get it. Thank you. There is a question from Kirill. I will read it. Paraspeckle proteins are often the same proteins involved in stress granule formation. Well, yes, and that. Might there any observable perturbation to the stress granules as well? Might there be some observable perturbation of the stress granules as well? Likewise, is stress granules assembly known to mediate the antiviral response of the cell? I think this is also a proper question. So, if this system can be extended from paraspeckle to stress granules.
So the stress granules are inside, they are in the cytoplasm. So I did not stain for them, but the majority of proteins that I showed here, for example, nono is not present in the stress granules, right?
No,
So I also didn't, I think FUS is, for example, present also, maybe or not. But most of them that what I was talking about, SFPQ, they are only in the nucleus.
Well, yeah.
So we did not look specifically on the stress granules. I tried to do solubility profiling to see if there are any other phase-separated organelles are affected. But there was no clear phenotype, and we did not stain specifically for stress granules. There still might be a phenotype because, of course, it's huge infections going on, and at some points viral proteins would be going to the cytoplasm after 8 hours post-infection, for example. But we thinking also that there might be at the beginning when we started working on paraspeckles, we actually had a different theory why it might happen because we at the first time we expected that the paraspeckles would be rather growing because we thought like maybe they would be sequestering viral proteins and kind of trying to counteract infection. Of course, it's also cell lines what we used. In the primary cells, non-transformant, we still see the same phenotype. But of course, we don't know how it will look in the proper organism where immune system is also properly present. We cannot exclude that there is still also immune response involved.
Yes, of course. Actually, it's a very elegant mechanism of hijacking. If this is conserved in, the organism, of course. I have the wonderful work by Kirill. Thanks. Kirill is happy about the answer, and I like to. So he's happy about this. So I want to, maybe I don't know if you could speculate. We have a couple of minutes left, so a little bit. So FAS is one of the proteins involved, and of course FAS is associated with ALS, so the neurodegenerative diseases, and often I read there is a connection also with with the beginning of these diseases. Do you think I don't know? Would you like to speculate about this. Well, but before I say something, some other people are asking things. So yeah, I want to know if you have a connection between things. If you see that if you in your experience, what you experience, or if you have made some, the idea of talking about stress granules is very interesting. Although, as you said, it's different compartments. But I was thinking whether, I mean, this can be extended to other processes that can be downstream of the viral infection.
I think here it's difficult to say because also we don't know the role of paraspeckles in the first place. So that's why it's so hard to speculate. Like, what are they actually doing? Because there is like in the non-infectious way, because we also thought like maybe because the reason we have the membraneless organelles sometimes is that we can either create an environment to do the reactions faster, the biochemical reactions inside the cells, or maybe sequester some, maybe these all splicing factors are stored with till the right moment. I think as for paraspeckles, it would be really interesting to know why we have them. Because I think it's only mammalian cells that have them. We cannot study them in simpler mechanisms like yeast, where it would be much easier. And I also I'm a bit fascinated by them, but I also would not like to know like what is their role.
Oh, yeah. So I think the secret
Um,
is in them. Understanding what is their function will be the key
yes,
to unlock.
yeah.
There is a question from Davide De Petitonelli, from my institute actually. So, hi Iulia, nice talk. Can you comment on the role of influenza A with respect to endoretroviruses, which are known to interact with paraspeckles?
Yeah, that's why we also at the beginning expected, because many other studies show that the paraspeckles either increase, on the other sides, they are either older and do not have proper quantification. That's why at the beginning, when we seen our phenotypes, it took us a while, first of all, to believe, does it really happen? Maybe we did something else. Maybe in this paper, they used FCS, they didn't use FCS, changed the infections media to see if it's really true that we have, because I think majority of papers that I've seen on other viruses, they say that paraspeckles increase.
Yeah.
And here we have, but I checked so many cell lines. I checked several strains of influenza. We never worked with different viruses. We know that it conserved the other strains, like also influenza A, pandemic variant, but also H3N2. They all disrupt paraspeckles. While for the other viruses, the phenotype was the reverse. I think it's interesting to investigate and to see, because also if influenza does something really completely different to other viruses, it's also not, would be interesting, then it would be something very specific for the virus.
Do you,
But we don't know.
I have a stupid question. So I think this is a very well response to Davide. Have you observed in the cell cultures if something odd happens to the cells in which the paraspeckles are disassembled, regardless of the spreading of the virus? Does it have any phenotype that you can report?
Actually not, because my knockout nanocells, like I passage them for quite long, I freeze them, I defreeze them, and they seems to be absolutely fine. They don't have paraspeckles. So I think the phenotype probably would be only visible in case if you have infection or some stress, because under normal conditions, they do not seem to need paraspeckles in the first place. At least I've worked with these clones, I mean, at least for 2 years, and I never noticed anything. They seem to be growing fine. They divided with the same, as the same as the wild type.
Yeah, so it's clearly something that has to do with the organism or more complex systems, perhaps neurons as well. So David is happy. You know if ERV changes special upon influenza disruption of paraspeckles. If it's quick, because I think we should move at this time. So endoretroviruses change their special upon influenza.
We never checked that, and we also never did any. I think we never did any how is it called, RNA-seq, to be able to just quickly check what is going up there.
Thank you. Well, I mean, it's a proper question. Thank you, Davide, and thank you, Julia. It was an amazing talk. I hope
Thank you.
that I can hear more from you. So now we have Giorgio Bini is a computational person, is also a recent postdoc because he obtained his PhD towards the end of last year. And he'll be presenting some work that actually comes from my lab related to RNA-RNA interactions. So Giorgio, the floor is yours. Have a nice one.
Okay. So first, thanks to the organizers and also to Gian for the beautiful introduction. So I'm Giorgio Bini and I am a postdoc at the Italian Institute of Technology in a group of Gian, Gaetano, Tartaglia. And my research focuses on the intersection between AI and RNA biology, where I develop deep learning tools to predict RNA-RNA interactions. And the work I did during my PhD has led to a paper that was recently published in Nature Communications. And today I will present the main result behind the project. So, as a quick introduction, RNA-RNA interactions mediate several functions in the cell. They play both regulatory and structural roles, helping to organize complex interaction network. And they are involved in key processes such as splicing, translation, RNA interference, transport, and stability. And overall, they represent a fundamental layer of gene regulation. So, key aspect of the project that I would like to highlight is the work we did on the experimental data, because a major limitation in the field is the lack of suitable data sets for training AI models. So, in close collaboration with my colleagues who have a strong biological background, we collected several transcriptome-wide techniques, and we can group these methodologies into main categories. So we have psoralen-derived methodologies such as Paris and Splash, which detect through UV cross-linking direct RNA-RNA interactions. And then we have protein-based methodologies such as RIC-seq and Mario, which detect RNA-RNA interactions occurring within ribonucleoprotein complexes. And we collected all this data and we then applied filtering and bias control in order to obtain a machine learning ready dataset of about 118 interactions of high confidence RNA interactions for long RNA molecules, which are the focus of our study. And we also released this dataset as an open resource. So once we detect RNA-RNA interactions, the question is what are the sequence features that drives them? And a striking patterns that we observed involve repetitive elements. So we know that repeats are highly abundant in the genome. It's estimated that more than the half of the human genome is composed of repeats. And there are biological reasons why these motifs are designed to be complementary to each other. And this is what motivated us in studying repeats in the context of RNA-RNA interactions. And in this slide, I show part of the amazing work that my colleague Adriano Setti did for this project. And here on the x-axis, we can see all the classes of repeats which differ for composition and length and function and location in the genome. And here on the y-axis, we can see all the analyzed RNA-RNA interaction dataset. And this analysis, which is called nucleotide level repeat enrichment analysis, the idea behind this analysis is to quantify the overlap between the presence of these repeat classes and the interaction binding sites detected experimentally. And as we can see here, there is a statistical significance of this overlap, especially for these three classes of repeats, which belong to the family of low complexity repeat that are strongly enriched across all the datasets. And this suggests that they play a fundamental role in the context of RNA-RNA interactions. We focus on low complexity repeat in the paper, and we also highlight that they act as sticky hubs, meaning that the low complexity repeat patches are involved in multiple contacts, involving also the presence of proteins. And recognizing the central role of sequence determinants, we then decided to develop a sequence-based prediction model for RNA-RNA interaction, and we called it RIME, which stands for RNA interactions model with embeddings. And the schematic overview of the model is represented here. So given two input sequences, we encode them with sequence embeddings from a language model called nucleotide transformer, and then we process them with a neural network to predict whether they interact. Here I go a bit more into the methodology behind our evaluation framework. So how do we model these sequences? We can see that this schematic representation, so two interacting transcript pair can be seen both as a contact map, where we have all the pairwise connections between the nucleotides, and here we have the interacting region highlighted in blue, or we can see as below with a sequence level view. And on the right, we have non-interacting transcript pairs that we obtained by randomly shuffling the RNA-RNA interactions detected experimentally. So here we face the computational problem, because since we were interested in long RNA molecules, these matrices can be very big, because RNA in our dataset can be long, like tens of thousands of nucleotides. So it is a computational problem to process them, and we tackled it by a window-based approach to study these interactions. And based on the region we sample, we can identify several cases. So for instance, in this blue window, we have a positive case because we are including the region of interaction on the two transcripts. And then we can have several negative cases. So for instance, we can sample from random region between two interacting transcript pairs, or we can do the same between two non-interacting transcript pairs. And then finally, we have this negative case, which is the most challenging, because here we have paired region observed in the positive set, but combined in non-interacting transcript pairs, meaning that these two RNAs were interacting with other partners in the dataset. So these are interacting prone regions, and as we will see in the results, this is the most difficult case for model prediction. So based on the different composition of the window that we sample, we can design an evaluation setup that allow us to approach RNA-RNA interaction prediction from two different but complementary perspectives. So we identified two main tasks. One is discriminating RNA-RNA patches, and one is discriminating RNA-RNA interactors. And intuitively, they answer to two different questions. So discriminating RNA-RNA patches answer to the question, where are these RNA interacting? Meaning that a tool which achieves a high performance on this task, it is able to discriminate interacting prone region from non-interacting prone regions. While in the case of discriminating RNA-RNA interactors, the question is, which are the interacting RNAs? So here we will test whether the model can be specific on the interacting binding partners. And here I show the results starting from the discriminating RNA-RNA patches task. And here we can see the results of RHYME, which is the blue curve, compared to the existing tools, which are mainly based on thermodynamic principles such as base pair complementarity and accessibility. And as we can see on both human and mouse, and also on both psoralen-based and protein-based methodologies, RHYME outperforms existing tools with improvements of up to 12% in ROC AUC. And these results suggest that these embeddings can capture complex sequence patterns that are difficult to model with traditional thermodynamic-based tools. And here we can see the results of the discriminating RNA-RNA interactor task. So although overall performances are generally lower, RHYME is still the best performing tool. And beyond performance metrics, our analysis made us to ask what are the factors that drive RHYME predictions. And in order to answer this question, we were interested in analyzing the relationship between these three entities. So we have the data quality, which can be evaluated by the number of chimeric reads supporting the detected interaction, and then the model confidence, which can be represented by the RHYME output score, and then the model performance that can be evaluated with, for instance, the ROC AUC. And here on the right, we can see an heat map with all the correlation between these pairwise comparisons in both psoralen and RIG-seq. As we can see, there is a high correlation score, suggesting the reliability of RHYME. And here in the next slides, I will go a bit more in the detail to understand how we calculated these correlations. So we start with performance and confidence. So here on the x-axis, we have the percentage of bottom and top predictions of RHYME based on its score. So basically the idea is that we can take the top 5% of positive prediction scores based on the output score of RHYME, and we can evaluate how strong they are over the available data set. And as we can see, there is a nice monotonic trend on both positive prediction and negative predictions. So evaluated by the precision score for positive prediction and the negative predictive value score. And this suggests us that the confidence of the model can be a proxy for its performance. And here we can see the relation between the quality of the data represented by the number of chimeric reads supporting the interaction and the performance, which is represented by the ROC AUC. And here we can see that on both psoralen-based and RIG-seq dataset, there is a nice monotonic trend on almost all the tools. And interestingly, RHYME, it's always the best performing tool. And finally, we analyzed the relation between the confidence of the model, which is represented by the RHYME score, and the quality of the data, which is again represented by the number of chimeric reads. And as we can see, there is again a nice monotonic trend, suggesting that we can use the RHYME output score as a proxy of the quality of the underlying data. And then, since we say that low complexity repeat play a central role in the context of RNA-RNA interactions, we wanted to assess whether RHYME could identify this interesting biological pattern. And here in this plot, we can see that when a low complexity repeat is present in one of the binding patches, the overall RIME score is generally higher than in the absence of low complexity repeats. And this trend is even stronger when two low complexity repeats are present in both interacting patches. And this is also quantified with a statistical significance across all the analyzed data sets. So since we know that in deep learning use generally a larger amount of data bring to higher performance, after validating RIME, we decided to train a final version by including all the available pseudorime-based data. And we then tested its performance on an orthogonal dataset such as RIC-seq. And we can see that on both the patches task and the interactor task, the new version of RIME outperforms the previous one. And beyond the performance metrics, we also wanted to assess whether RIME could capture high confidence interaction coming from high confidence techniques such as comrades. And here, as a case study, I show the long non-coding NORAD, whose dysregulation has been associated with cancer. And here in the top panel, we can see the experimental signal from the comrades experiment, where it detects all the interactions of NORAD with its multiple targets. And here in the bottom panel, we can see the RIME predictions. And as we can see, RIME correctly identifies the same interaction hotspots along the transcript. We then moved to some targeted pull-down experiment. So here, the author were interested in the long non-coding tinker experimental detection, and what they observed is that this validated interaction, it's proved to regulate the level of this tau1 protein. And interestingly, RIME correctly identifies this interacting patch, suggesting that the tool is also able to detect functional RNA-RNA interactions. And as a further evidence, we also tested over this high-quality targeted pull-down experiment, where the authors proved through a deletion that this interaction between the long non-coding SMART and the messenger RNA of the MLX gamma gene directly regulates the level of the protein of the MLX gamma gene. And this interaction that has been validated, it's also detected from RIME. And interestingly, RIME also is suggesting that there is another interaction close to the 3' UTR of the MLX gamma transcript. And these findings is also consistent with what the authors found in the paper. So to conclude, we analyzed several transcriptome-wide techniques, and we collected them, and by applying filtering and bias control, we released a benchmark dataset specifically designed for machine learning. And we then, building on this, trained a deep learning framework for sequence-based prediction called RIME, which outperforms existing thermodynamic-based tools. And RIME also excelled at predicting high-quality interactions. And we then revealed biological insight in this field, highlighting the role of low complexity repeats in these interactions. And we confirmed their importance through both enrichment analysis and computational predictions. We also released several open resources such as the machine learning dataset under Zenodo, or the rhyme code, which is available on GitHub, and then also a web server under Tartaglia Lab with a easy to use interface. And with this, I would like to thank all the people involved in this project and all the people of my lab, and also you for your attention.
Super. Thank you very much, Giorgio, for the presentation. I see already two questions in the chat, the question and answer chat. The first question is, from Fabrice, does the dataset include only perfect matches between pairs of transcript sequences, or are there any pairs with mismatches? And then, is there any potential bias in the predictions, top or bottom, based on the ratio of perfect and imperfect matches in the datasets? So it is two parts. So the first part is if you just have perfect matches between pairs of transcript sequences.
Um, I'm not sure I understand what you mean with perfect match. If it's related with, for instance, canonical base pairing or non-canonical base pairing. Yes, we do have also non-canonical base pairs detected experimentally. And also a possible source of mismatch is related to the fact that some interactions are detected by different chimeric reads and they may overlap. So what we did in that case is to build a bigger interacting region in order to have uniformity in the data, which includes both evidences from the two chimeric reads. So maybe this.
I would say that this could be exactly the answer.
Okay.
If there is anything to clarify, please do it in the chat. I'll be in this question and answer. I will be happy to inform Giorgio. But I suspect the point is exactly about perfect base pairing. So so the second part of the question, if there are any bias due to the imperfect perfect matches, basically you answered already. But if you give it some thoughts, I read also the other two questions, so then maybe some say arguments can be used for killing more than one pigeon with the same stone. Beatrice Corradi, hi Giorgio, very interesting talk, thank you. Just a curiosity, when selecting the length basis for the RNA interaction patch, is there any sequence composition or overrepresentation to avoid? Thank you. So if length and composition might be a bias or anyway are important in your statistics.
Yes, thanks for this question. I will show a slide. Sorry, I think. Can you see the slides?
We see the last one.
The last one,
I see
okay.
that you are searching. Yeah.
Yes,
Now I see.
searching a slide, which is not this one. Yeah, this one, which is about the composition. So there is an unbalancing composition in the experimentally detected RNA-RNA interactions, and actually this was also the most challenging part of the project because we had to balance all these properties that are biological evidences of the data. So for instance, we can see that CDS 3' UTR is strongly enriched in the dataset, and our effort was to build a negative set that balance all these properties across training, validation, and test sets. And also, I mean, this is also related to other problems like here, for instance, in distributions, um, in this density distribution, I show that if we pick randomly one RNA, what is the probability that it appears in the positive or in the negative set? And we are sure that this probability was on average 0.5 across all the possible negative classes. So all these elements ensure the unbiased composition between the positive and in the negative set to avoid biases from the learning phase.
Beautiful, thank you. I will read the other questions now. Bianca Perattini, oh, it's a long time since we haven't spoken. So Bianca is a dear friend. Giorgio, great talk. Your results seem to suggest that these RNA-RNA interactions might not depend on stable secondary structure, if I correctly understood. Do you think this should change the way that we model RNA structure interaction in the cells? And how do you differentiate biological and meaningful interactions from promiscuous pairing due to sequence repetitiveness? So in a way, it's connected to the first question, I would say. So again, are these interactions stable enough? And what is the connection between simple repeats and promiscuity in your view?
Yeah, also the point I think interesting is about the secondary structure she was mentioning. We also tested actually RIME over secondary structure prediction just to see if there was an overlap between the performances, an enrichment in the performances also for this task. And I have also something here. So here we were trying by analyzing these comrades experiments, we also detected some patterns in the secondary structure prediction of NORAD. And here we can see that there is some signal of the RIME predictions with respect to the experimental signal, which maybe it's more clear here from this slide where we have that in correspondence with higher RIME-full score, it's corresponding in a general higher number of chimeric reads supporting the intramolecular detection, experimental detection. So in general, I would say that our results suggest that RIME prediction are strongly correlated with the quality of the data. And this is true on both intermolecular and intramolecular predictions. And I wouldn't exclude that. So this suggests that RIME also captures some pattern useful for intramolecular prediction. So yeah.
Beautiful. Very last, because we have 2 minutes to
Okay.
finish. So if you manage, wonderful work. This is from Kirill. Can the tool also be used to explore potential small RNA interactions? Small RNA, I guess, below hundreds.
Thanks for the question. No, I would say no, because we focused only on long RNA interactions. Actually, there are other deep learning framework designed for microRNA related interactions. And while there were not approaches based on deep learning for long RNA interactions, and the overall study focus on long RNA interactions because also of the biological mechanism behind, they are different, and I would probably expect some signal also on microRNA interaction, but they were not included in our training data.
Beautiful. Thank you for your question. For your answer to the questions. Maybe in the future can be improvements, I guess.
Yeah.
And thank you, everyone. And so David also is thanking you and thanking Iuliia. So I also personally want to thank both of you for this, and also the organizers for this beautiful opportunity to present our work. So thank you everyone. Have a nice rest of the morning, the afternoon, wherever you are. Thank you.
Thank you.
Bye.
Thank you.